Hi, welcome to Chapter 3 review. Okay, these are the 4 biomolecules, bio meaning life, and molecules we've talked about, combinations of elements and atoms. So we have carbohydrates, we have lipids, we have proteins, and we have nucleic acids. So let me get the pen going here. So these are the 4 major categories. Then they give you some examples here, and then they tell you the subunits. So I want to point out the subunits because these are the terms that we sometimes get confused about. So saccharides, if I take saccharides and put them together, so link them together, that'll give me a carbohydrate. If I take fatty acids and link them together, that gives me a lipid. If I take amino acid 1 plus amino acid 2 plus amino acid 3, and I put those together, that'll give me a protein. And a nucleotide, so one nucleotide plus another nucleotide will give me a nucleic acid. So sometimes you'll see the word monomer and polymer. So mono means one, poly means many, right? So in this case, this is the monomer. And if I take a bunch of them and put them together, I get a polymer. This is a monomer. If I take a bunch of them and put them together, I get a nucleic acid, which is a polymer. So we're going to look at these 4 individually and learn a little bit more about them. In this first couple of slides, I'm showing how you can put them together and pull them apart. So if you want to put 2 monomers together to make a polymer, you have to do a very specific reaction. So in this case, dehydration, and it'll be better when we look at the actual chemical formula here. So the dehydration reaction, if I want to make polymers from monomers, that means M1 plus M2 plus M3 will give me a polymer. So synthesize means make, right? So if I want to take monomers and put them together to make a polymer, that's called a dehydration reaction. Hydrolysis splits polymers into monomers, and hydro means water, and lysis means split, right? So this would be if I have a polymer and I want to break it up into individual monomers. We do have to have something called an enzyme in order for this to happen. An enzyme basically makes the reaction occur. Go away. All right, back to the dehydration hydrolysis just so you can see what I'm talking about in a little more detail. So dehydration. I have monomer 1, and this is 2 monomers that are already stuck together. So these, I want to add this to this group. If I take water out, that is called a dehydration reaction. I went from 2 to 1, right? I stuck them all together. Now if I want to do hydrolysis, that means split with water. I have to take water and split, and now I have 2 separate pieces from the one. Again, this should, when you see the water, you should realize that water is important for this step. If you do not have water, you're not able to do dehydration or hydrolysis reactions. So this is yet another example of how water is a very important biomolecule. Okay, I'll tell you just a quick little story in case you're having troubles with this. What's really interesting about dehydration and hydrolysis is we don't think about it in terms of food all the time, but the ability to be able to store energy. So let's say let me clear all this. It's late at night and you decide you're going to go to a McDonald's and you're going to get a Big Mac and fries, but you're not doing anything, right? It's 10, 11 o'clock at night, you eat the food, you decide you're going to go to bed. So you've got these food molecules in 2 separate pieces, right? You're going to pack all your food together and you're going to store it. We can store some of it in our liver. We can store it as fat if we need to. Well, let's say you get up the next morning at 6 o'clock and you're decided you're going to go for a run. You're going to go for a 5-mile run. You don't consume anything. You don't eat anything. You don't drink any water. You don't eat any food. You just throw on your clothes and you go outside. Well, this food that you stored the night before, we take that food and we break it down into individual pieces, and then we can use this to make energy. I mentioned ATP earlier when we take glucose and we turn it into an energy source that we can use, and that's called ATP. So without dehydration reaction and hydrolysis reaction, you would not be able to store food. Anytime you wanted to do any kind of activity, you would have to consume food before you did the activity. So we are using both of these reactions every second of every day, and we don't even know they exist sometimes, right? Okay, so let's talk about carbohydrates. The one sugar is called a monosaccharide. So glucose is the monosaccharide that we talk about the most. Glucose is what we use to create that ATP. So we use, hello, I don't want yellow. So we use this to make ATP, right? When they say isomers, they're saying this chemical right here, that makes up glucose. If we take 6 carbons and 12 hydrogens and 6 oxygens and put them together, we call that structure or that molecule glucose. Well, fructose and galactose look a lot like glucose. They're just rearranged a little bit. They still have 6 carbons, 12 hydrogens, and 6 oxygens. They're just not all in the same exact location all the time. So in this example where this OH group is, or where this group is, it might be different on fructose, galactose, and glucose, but they all have the same chemical formula. We'll also talk about ribose and deoxyribose, which help make up nucleic acids. So they're sugars that help make up our DNA. One of the things to notice too is, what does every sugar end in? It ends in ose. So anytime you see an ose, you should be pretty sure that that's a sugar or a carbohydrate. So what can we do with simple sugars? If we wanted to put them together, we could link them together. So here is one sugar, and they're showing you in the dehydration hydrolysis reaction. So here's one glucose plus another glucose, and we're going to link them together to have something called a disaccharide, or when we take 2 monosaccharides and put them together, we call it a disaccharide. So now there's something called maltose if we take 2 glucoses and put them together. One that we're probably also familiar with, if you take glucose and you add a fructose to it, you will get sucrose. And sucrose is table sugar. You sprinkle on your Rice Krispies. There are also some more complex, and we call those polysaccharides. So we saw monosaccharides, disaccharides, and now polysaccharides. Polysaccharides are what you would think of if you talk about starches. So you can see that these are a lot more complex. It's not just 1 or 2 sugars, it's many chains of sugars. And sometimes the chains form these sheets, as you can see. So this one is important, glycogen. Ooh, I'll put the line through it. Glycogen. Glycogen is how humans store glucose. So humans store glucose as glycogen. Starch is how plants store glucose. Cellulose, also plants. And this is usually undigestible. This is what we would call fiber. You can't really digest it or break it down. We can break down starches, but we can't break down celluloses. But why is fiber good for you? Fiber is good for you because it cleans out your intestines as the fiber is coming out. So any buildup can be removed by consuming fiber. All right, next are lipids, most commonly called fats, right? We're going to call them lipids. So here's a picture of a triglyceride, and I'm sure if you look at the picture for a second, you can see why it's called triglycerides. There are 3 of them, right? 1, 2, 3. And these are fatty acid tails. So fatty acids. And that you can see their tail-like structure. So sometimes you'll hear the word saturated versus unsaturated. So here's unsaturated. It's saying not every hydrogen. You see how the hydrogens are missing? That carbon has formed this double bond. And in this one here, this would be an example of saturated. Do you see how every carbon has 2 hydrogens attached to it? So saturated fats lay flat and they stick together. So think of like butter. Unsaturated fats, they have this little kink in their tail because of this double bond. So think of oils. So this is a liquid and this is a solid. So that's why sometimes they say that unsaturated fats are better for you because they're in liquid form, so they don't pile up. They don't stick together. They don't stick on the inside of your blood vessels. There are many different types of lipids, so we're going to talk about the ones that we need to know about. 1, triglycerides, saturated and unsaturated fats. And then the next here's another example, just giving you more details about saturated, unsaturated, and trans fats. Again, trans fats, the hydrogens are on opposite sides. This is not really the best. Trans fats, they get a bad rep because they're chemically modified. They're found in processed food, and they're not easy for our bodies to break down. So they hang around a lot longer than saturated or unsaturated fats. They've been artificially manufactured, if that helps. All right, so these are other types of lipids. So other types of lipids that we need to talk about because they're important. So phospholipids, I'm going to show you a picture in just a second. This makes up our cell membranes or plasma membranes. So if I have a cell, it has this outside layer, and this is called the plasma membrane. And this decides what can get in and what can get out. So it's very important to the cell. It's like the fence around the cell. And it's made up of fats. We also have something called steroids. This is where we get our sex hormones from, testosterone and estrogen. They are steroid, they are fats. And then some waxes found in plants and animals, used as waterproofing. You can think of maybe earwax. On plants, like the outside of grapes or leaves. They help keep the inside moist and juicy, if you want to think of a grape, and then the outside's the outer coating that you have to bite through to get to the inside. So why are all of these different lipids mentioned? I want you to see how diverse lipids are. A lot of people, when we talk about fats, they think fats are all bad. Well, sex hormones, we need testosterone if you're going to become a male, and you need estrogen if you're going to become a female. And both of these are very important in sex drive, in the ability to procreate or have children. You can see here phospholipids are important for the outer membrane of a cell. If you didn't have a plasma membrane, you wouldn't have a cell, period. So fats kind of get a bad rap. We need to understand that lipids are very important and they're vital, and we need them on a daily basis. So here is a phospholipid. What do you notice the difference about a phospholipid and a triglyceride? So phospholipids have 2 tails. Oh, sorry. And then our triglyceride had 3 tails, right? So it's just a modified version of a triglyceride. And I wanted to show you what the plasma membrane looks like. It's the head of the phospholipid and then the little tails. So basically what they're trying to show you is this is called a phospholipid, bi meaning 2, layer. So you see how there's 2 layers? Basically these tails come together and the heads, one stays towards the inside and the other towards the outside. And we'll talk more about these as we move into chapter 4, I believe. But phospholipids are important in creating this plasma membrane around every cell, critical for the cell to live. All right, the next are proteins. So what are proteins? Proteins are, first of all, they're one of the most versatile of life's biomolecules. They do all sorts of functions. So they're for support, for metabolism, for transport, for defense, for regulation, for motion. So we have, you can see in these pictures here, they're giving you muscles, that's a protein. We have something called myosin and actin. These are proteins that are made by muscle cells, and they enable your muscle cell to contract or generate force in order for you to walk, talk, run. Hemoglobin is a protein. Hemoglobin is what oxygen binds to. Hemoglobin allows you to breathe in oxygen and transport that oxygen all over your body because your muscles need oxygen. I'll put it in here because I think it's interesting. Most people don't. We're going to talk about it down the road. Glucose and oxygen turn into carbon dioxide, water, and energy, or ATP. This is called cellular respiration. So why do we breathe in oxygen and why do we consume food in terms of glucose? Because we're trying to make energy so that we can do this. Hair is a protein. So we have lots of proteins that carry out various jobs within the body. Here are some of the amino acids. Remember, amino acid is the monomer. So amino acid is the monomer, and when you link them together, you make a polymer, which is called a protein. So if I take amino acid 1 plus amino acid 2 plus amino acid 3, and I get out protein A. What type of reaction is responsible for adding monomers together to make a polymer? That would be dehydration reaction, right? So, showing you how amino acids are joined together, and I've already mentioned dehydration, so I'll let you look at that slide. I won't go over it in more detail. One of the things that's important about proteins is its structure. So proteins, sometimes to carry out a specific job, they have to be in a very specific shape. And we'll look at some of these shapes in just a second, but one, I want to tell you that if the shape changes or the shape has been denatured, the protein will no longer work. So I'll just give you a quick little example. So let's say you have a protein that has to bind an item, and that item is, we're going to say this is sugar. This is sucrose, and it's a square. And this will bind to this protein, and the protein will cause it to split into two smaller pieces. Remember that glucose plus fructose equals sucrose. So let's say we want to get just the glucose or just the fructose. The sucrose will bind, and you see how it's a perfect fit. The square fits into this protein, and it causes it to split into glucose and fructose. Well, what happens if the protein changes shape? What if the protein does this? Can the sucrose fit into that wedge? No, it can't. So you will not get a product out. So what am I trying to say? If you change the shape of the protein, it may not be able to do its job anymore. And we call that denaturation. Let's look at some of the organization. There's a primary, secondary, tertiary, and quaternary structure. And it's probably best to look at the pictures here. So what they're trying to show you is the very beginning. So when we first make a protein, it's just in this string form. It's just a sequence of amino acids. We call it primary structure. Now we can do some interesting things with that string. We can twirl it into a ribbon. We call that an alpha helices. Or we can bind it together in a sheet-like form, and that's called a beta sheet sometimes. Or this one, it just says pleated sheet, but it's usually called a beta sheet. So you have alpha and beta. These are secondary structures. Tertiary structure is when we take all of these and put them together. So do you see how there's an alpha helices, and then there's a string? That's the primary. And then over here there's a beta sheet. So I've taken primary and secondary and put them together, and that gives me tertiary. Now quaternary structure, basically if I took this, it's one of these. So quaternary is if I take multiple tertiary and put them together, now I have quaternary structure. The ones that fall into this category, there are very few. There's hemoglobin, collagen, which is in your skin, helps keep your skin elastic. So those are quaternary structures within the human body. All right, so we're on the last category, which is a nucleic acid. A nucleic acid, remember we talked about monomers? Monomer of a nucleic acid was something called a nucleotide. So I put one nucleotide so you could see. It's a sugar, a base, and a phosphate. That's one nucleotide. Now if I take a bunch of these nucleotides and put them together, I end up with a nucleic acid. We call this deoxyribonucleic acid. This is DNA. DNA is going to copy itself into RNA, and RNA is going to make proteins. It's the code to make proteins. So our body decides, our cells, our DNA decides which proteins it's going to make. So here's another example of DNA, and they're showing you the nucleotides as they've been strung together. So remember, a nucleotide is a sugar, a base, and a phosphate. So this is nucleotide 1. Now we have a sugar, a base, and a phosphate. This is nucleotide 2. Excuse me. And when we link them together, we get deoxyribonucleic acid. And these are many different ways that you can look at it. Here it is in an alpha helices, and this is a space filling model. So this is what I just talked about, and we will talk more about this as we move through this class. But basically DNA, which has deoxyribose, that's the sugar, right? And then RNA, which has ribose as a sugar. So this is sometimes called the molecular kind of dogma, how we make proteins: DNA, RNA, and then proteins. What's different about ribose or RNA is that it has a base that DNA doesn't have. So this is uracil, and I know this is RNA because it has that uracil. Go back to DNA. Do you see any U's here? No, we only have, come on, behave. We only have T's. That is thymine. So DNA always has T's and RNA always has U's. That's how we can tell them apart. So you should look at kind of a comparison, and the book does a great job of showing you the difference between RNA and DNA. So when you look at these, what's the difference? Well, the sugar for DNA is deoxyribose. For RNA, it's ribose. The bases for DNA are A, T, C, and G. Some people like to say G-cat because they can remember that. I don't care how you remember the bases. And then for RNA, it's A, G, U, C. Strands. DNA is usually paired. You know how we saw the two strands and they were connected, kind of like a ladder, where RNA is usually just one strand with the bases on it. DNA forms a helix. We'll take this ladder and we can twist it. RNA is just one single strand, so it can't form that helix. So here's an example of a mutation. I just wanted you to see how important it is to make sure that the amino acid sequence is correct, and that comes from the DNA sequence, right? So in this particular case, they're trying to show you these are amino acids. And remember, our DNA, nucleic acids, programmed what our protein was going to be. So if there's any sort of mutation, if there's something wrong here, it will cause something to be wrong here in the protein. So these 4 amino acids, if you link them together, they're going to give you a normal red blood cell. If you were to switch these 2 amino acids because somehow your DNA code was wrong and that led to an incorrect protein code, and you get this amino acid in the 3rd sequence, it causes your red blood cells to form this sickled shape. So one mutation in your DNA can cause a mutation in your protein, which can cause your protein to have a different shape, and this is really non-functional. What will happen to these sickled red blood cells? Remember, your blood vessels are round. Well, that's why the blood cells are round. Well, what happens if in your blood vessels you put sickled shaped red blood cells? They can become caught and clogged, and they can cause a lot of pain. The last thing I want to just mention is ATP. I've talked about ATP a couple of times, but I want to make sure we realize ATP really falls under the nucleic acid category. So this is a sugar with a base attached, and it has 3 phosphates. Remember, DNA only had 1. Remember, nucleotide had a sugar, a base, and a phosphate. Well, ATP is a sugar, a base, a specific base though, adenine. So that's the only base that's there. And you don't have the other bases, you just have the A. So there's a base, there's a sugar, and there's 3 phosphates. And inside these phosphates, these are all negative. So when you take negative items and you try to shove them together, they try to repel each other. And in order to get them to link together, you have to put in tons of energy. So sometimes people think of, let me just erase some of this. Sometimes people think of these as a coiled spring. In order to get the phosphates together, you have to push them together, and you have this coiled spring. Well, what happens when you cut the spring? When you cut the spring, the P goes flying off, but you get all the energy that you stored as you tried to mush these 2 phosphates together. So basically, this is what ATP does for us. We take glucose, and we cut down the glucose. We get the energy out, and we store the energy in these bonds between the phosphate, and then the ATP can go anywhere in the body, and when it gets to the body, it breaks that bond and it releases energy. We'll talk more about this as we move through Unit 2. I just wanted to give you a brief update of what's to come, right? Okay, last slide. Just making sure that you're connecting all these concepts. There are 4 types of large biomolecules that are present in all cells. So make sure that you can list what those biomolecules are. We have our carbs, we have our lipids, we have our proteins, and then we have our nucleic acids, right? Make sure you can give me information about each one of these. DNA, it's the genetic code that determines what type of proteins can be made. What is DNA? Remember, DNA is a nucleic acid. DNA is going to copy itself into RNA, and RNA is going to be the code to make all of the proteins. Mutations are a source of genetic variety for evolution. I showed you an example of a bad mutation, but that mutation for sickle cell anemia actually prevents the red blood cells from being invaded by a pathogen. So malaria, the pathogen responsible for malaria, doesn't like sickled red blood cells. It doesn't want to infect them. So in some cases, if you lived in an area where malaria is prevalent, having sickle cell wouldn't be so bad. You actually wouldn't get an infection, and you wouldn't die from that infection or become very, very ill from that infection. But when you live in an area that doesn't have malaria, that's not, the benefit doesn't outweigh the illness. So not all mutations are bad, sometimes they're good. And ATP has the same structure and function in all cells. Remember, ATP is also a variation of a nucleic acid. ATP can be found even in bacteria. All living cells use ATP for energy, to carry and shuttle energy throughout the body. Well, I hope I've made a little bit of sense of Chapter 3. Please email if you have any questions or comments or concerns. I'll see you guys Chapter 4. Bye.